One Health and Plastics, a global experiment.
If anyone placed bets on the New Zealand trifecta, kia ora, tūmeke, tēnā koutou, tēnā koutou, tēnā koutou katoa, you have won. 3 New Zealanders in a row, isn't that amazing? Today, yes, my name's Jack Rivers-Auty, and today I'm going to be talking about the global experiment, and this experiment is plastics in the wildlife. I'd like to thank a lot of people right at the beginning. There's the list, but there's many more, but I just thought I'd start at the beginning by thanking that I'll be presenting a lot of work from my PhD student, Alex, up the back, and a number of other people. I did want to talk about my journey in One Health, just to start with. This is my career along the x-axis. This is the proportion of my research that's One Health. Started out very human-focused. Then I looked at diet and zinc deficiency and how that impacts inflammation in human health. But that's very linked to soil health and animal health because we consume animals and that's where we get our zinc from. So if they're zinc deficient, we become zinc deficient. And then I moved on to plastics research and I'm researching wildlife and human health. And I felt like I became a fully fledged One Health researcher. But my awareness, my knowledge that I am a One Health researcher follows this graph. It's almost a straight flat line along the bottom and then it spikes up. And this spike is thanks to this lovely man over here. About a month ago, 2 months ago maybe, I went to his talk and I realized that I'd found my people. He articulated my research experience all in one beautiful talk. And he gave me a wonderful weapon. The Tasmanian School of Medicine has been pestering me for researching birds and not humans. And I felt like I always struggled to communicate the link between this. But this wonderful term, One Health, and this wonderful man, Nicholas, has helped me communicate to my department how it's all related, right? It's all linked and all tied together. Now, my research is focused on this. Many of you will have experienced this swollen ankle. That's inflammation. And I explore inflammation from the context of it being a good process, but also it sometimes being a bad process. You all probably would put ice on that to try and slow down that inflammatory process. There are many ways inflammation can be a bad process, but one of the cleanest and most simple examples is your immune cells rush into this joint here and they start to produce bleach. And I don't mean a chemical like bleach, I mean exactly bleach. In fact, my colleagues over at the University of Otago, Professor Mark Hampton and Dr. Nick Magan, they actually use Genola from the supermarket as their standards because they found it's highly pure bleach and it matches the bleach that our immune cells produce. Now, they produce it for the same reasons we bleach our toilet, and that's to kill the germs, right? So that's a good thing. But you can imagine bleach in your joint could be a little bit of a bad thing, or bleach in your brain could be a bit of a bad thing, especially over a long time. So I explore how the inflammatory process can be damaging, and that's been virtually my entire research career. Now, we're going to talk about plastics. Now, it's always a grim thing, but I think everyone's now much more aware of the problem of plastic. It used to be that I had to introduce you that you've been exposed to plastic, but now you can't pick up a newspaper without finding out a new thing that is full of microplastics, be it your testicles, your breast milk, or the womb is full of microplastics. So I thought I'd just give you some homework. I want you to go home. I want you to shut the curtains. I want you to turn out the lights and then just crack the curtains a little bit. Then I want you to walk over. 80% of our clothing is made out of plastic. So I want you to walk over with your synthetic clothing and I want you to just tap it and watch the puffs of microplastic be released from your clothing, right? There was recently actually a study out today said we breathe in 68,000 microplastic particles a day. And a single shake of a jumper can release about 7,000 microplastics. So it's certain that we're exposed to microplastics, but what the heck is it doing? We don't know, and that's what we're trying to figure out. Now, I look at this guy. So imagine your immune cells. They're full of these receptors designed to detect things, detect pathogens, detect parasites, viruses. We also have receptors that are designed to detect other things. And this guy is famous. This receptor here, NLRP3, is famous for detecting particulates. It's really good at detecting particulates in your immune cells. And it's been associated with detecting the particulate uric acid crystals, which build up in gout, but also silicosis dust, which is a mineral dust that people breathe in when they chop kitchen benches, for example. Asbestos we're all aware of, the cholesterol crystals that build up in your blood vessels, even protein aggregates that build up in our brain can act like a particle and cause dementia and Alzheimer's disease. But does it activate in microplastics? And this has been one of the questions of my research. Can microplastics activate this receptor and induce an inflammatory response? So I actually came up with the idea to research this after watching a David Attenborough at 1 a.m. at a conference. I couldn't wind down, so I watched a David Attenborough. And he said, we didn't know what it does in the body. And I was researching all these other things. And I thought, I bet you it activates this receptor. So that Friday, I did an experiment. Now, this is such a Friday afternoon experiment, not high quality at all. But I took some immune cells, and then I took a Swiss Army knife that came free with a pipette I'd bought, and I filed a pipette tip box. And then I cleaned it with ethanol, the plastic, and then I put the plastic on the immune cells to see if it would induce an inflammatory response. And this is the result I got. It's not really publishable because I don't know how replicatable it is, but this is the result I got. So silica is the positive control there because we know silica activates this inflammatory receptor. And there's the microplastics. And there's the immune cells that have had that receptor genetically deleted. So we can see by genetically deleting that receptor, we delete the inflammatory signaling. So we know that the plastics were working through that receptor there. And then I did some experiments that now I don't feel that great about. I did animal experiments where I took mice, I fed them plastics, I made them breathe in plastics, and I injected plastics, and I looked at the inflammatory response. And not surprisingly, we got a strong inflammatory response. So this is the immune cells present under the skin of a control mouse. This is the immune cells present under the skin of a plastic injected mouse. That was sort of the first experiment. So you can see all those immune cells rushing to the sites, and the immunologists amongst you will notice the shapes of those nuclei and recognize those are the damaging bleach-producing immune cells called neutrophils. But lab mice are kind of abnormal, and this is where I've started to grow as a researcher. Lab mice, we grow them in very sterile conditions, not quite sterile, but very clean conditions. They have weird microbiota. They have very weird immune systems. So it's not an ideal setting to do some of this research. They are very abnormal. Imagine we grew up in a bubble, sterile. We would have a very weird immune system and immune profile and bacterial profile. Also, I was injecting clean, naive plastics, and that's not the plastics we get exposed to. So over here, I've got the same inflammatory response in blue to naive, clean nylon. And then I've got some ground up environmental plastics. These are actually microplastics harvested and cleaned from the ocean. And we can see that they're more inflammatory. And my current PhD student, Oshin, has done heaps of microplastics research and shown that different colors are inflammatory. Different sizes and different shapes affect the inflammatory properties of the plastic. We know that plastics accumulate heavy metals and toxins while they're in the environment. So there are many, many ways that just putting naive plastic into a weird mouse is not the best way to research it. So I got the opportunity to research wildlife. Now, I want you guys all to imagine you're on an animal ethics committee. You're on an animal ethics committee and a project comes across your desk. And this is the project. I'm going to feed millions of animals plastic until they die, some of them. Some of them are going to be fed plastic until they die. And then I'm not going to measure anything. That's my project. What do you guys think? Do you think that's a good animal ethics project to come across your desk? We're not going to find out anything. We're not going to discover anything. All I'm going to do is do this experiment where I inflict plastic on animals. You could also sub in global warming. You could sub in air pollution. Does it feel like an ethical experiment? And it doesn't, does it? But we are already doing this. That is the global experiment. We are currently performing a global experiment on us with our synthetic clothing and on all wildlife with the plastics going into the ocean and going into the air. But also on top of that, you could put global warming, you could put car emissions, you could put novo chemicals. We seem to be inventing new chemicals all the time, and let's just go use that. That sounds like fun. Or farm runoff, pesticides, you name it. So we're doing this giant global experiment on billions of animals, and then we're spending a fraction of a penny to research the potential outcome of that. You know, if we were to imagine, this is actually my wonderful PhD student, Alex, went out and collected all the major public funding data on the research grants that were successful and research which percentage of it is looking at the health impacts of all these global experiments that we're currently doing on wildlife. And it's a fraction of a percent is where that funding's going. A lot of the funding is going to those previous experiments I talked about, the mice with the weird immune systems and those kinds of experiments. So let's go talk about a real model. What's a real environmental model that we can talk about? This is the Tuanui, or the sable shearwater. It lives on Lord Howe Island and it eats plastic. And I came to Tasmania and I looked at fellow plastics researchers, and I found Jennifer Lavers here. She was at IMAS at the time. Definitely, I would say, the most wonderful and greatest scientist I've ever met. I cannot say more about how awesome Dr. Jennifer Lavers is. She'd been studying these birds for about a decade, but she hadn't been looking at the deep physiology, the inflammation, all that kind of stuff that I was an expert at. So I kind of pitched her, hey, I can come help you with your research. And she very generously let me come along, basically. It was amazing to go research what are the potential consequences of these birds eating plastic. Here's Alex. So some of these birds, this is a tough slide. I was going to actually bring this up with Emily. It's like sometimes engaging in nature is not good for your mental health. Sometimes it is very difficult. And we all suffer from eco grief when we go out to this island, Lord Howe Island. So these birds have eaten plastic, sometimes to the point that they can barely function, but everything in their DNA says that they need to fly to the coast of Japan. So if they can't fly, they'll walk. And then if they can't walk, they'll try and swim, and then they'll get washed up. And so we come out in the morning as soon as the sun starts to rise, and we find these birds washed up on the beach who have tried to get to Japan, because that's everything that their DNA is telling them to do. And some of them unfortunately die, and some are in such a condition that the most ethical thing to do is to euthanize them. And then we can explore the physical and anatomical and physiological consequences that are happening in these birds. So this is the lab. There's the one called Jen. I probably should have put a more in focus photo. She's a bit Bigfoot at the moment. Blurry, just say out of focus. There's Alex. This is the lab conditions. We're not wearing gloves because we don't want to contaminate our samples with plastic. And that was unsettling to me as a biologist who worked in human labs coming out there, but totally makes sense. I'm just going to show you the worst bird. It is a graphical scene. So I'm going to show you a video of one of the worst birds that we had. So this is one of the worst birds that we have. Now, these kinds of birds aren't the best to research because it's so extreme. And so that kind of level of plastic is so extreme that it's going to cause such a range of physiological problems that these aren't the ones that we try and focus on when we're trying to do some of our more deeper and more complex biology. We certainly would take those samples and show what it does, but you can see there that it just keeps going. And it can take hours just to count the plastic at the end of the day. It's such an exhausting time. All my medical colleagues go, oh, how was the holiday? And I go, it was 16, 17 hours a day of this kind of stuff. So certainly not a holiday going to Lord Howe Island and looking at this kind of stuff. Yeah, and you'll recognize some of that plastic. One piece of plastic we found on my first trip out actually was a little Lego astronaut. And it was only produced between the years 1970 and 1975. So it may not have even been the first bird that piece of plastic had killed, you know, because the bird would rot around it and the piece of plastic would continue on. Yeah, so this is the huge amounts of plastic that we get from some of the birds. But what have we learned? What have we learned? This is Alex's favorite bird. What have we learned? Well, the first thing that we learned, this is actually the first bird I opened up. And I opened it up and I said, whoa, that stomach's inflamed. And they said, how did you know? Because they are environmental biologists and they are not, pathophysiologists, but that is shiny and puffy. That's a guaranteed sign of inflammation. The shininess is the fluid rushing in there. The puffiness is all the immune cells. So that's a highly inflamed stomach. And you might think, oh, the plastics are poking the stomach and that's causing inflammation. But here's some squid beaks. Now, some of these squid beaks are so sharp that they could cut your skin. But we don't see the same inflammatory response to the squid beaks. That looks fairly normal, that squid beak compared to that guy. So it's something else going on. It's not just the pokiness. These stomachs are designed to handle pokiness. And it's something unique to the pathophysiology of plastic. So we did what's called a trichrome stain. Now, a trichrome stain has blue in it, bright blue. The blue is scarring, right? And it's connective tissue. So this is a normal amount of connective tissue. This isn't scarred. All your cells need to attach to something really good. And so this blue is all the things that it has attached to. This is a bird that had a mild amount of plastic. And you can see that blue has really thickened up. That's scarring. So this animal has really scarred stomach. And we define this because asbestosis is the inflammatory scarring condition you get from breathing in asbestos. Silicosis is the inflammatory scarring condition you get from breathing in silica. Cirrhosis is the scarring condition you can get from drinking too much whiskey. Plasticosis is the inflammatory scarring condition you get from being exposed to plastic. So we define this new disease. But we also wanted to look at the milder cases. So we can take the birds and we can fill them up with water through a gavage, and then they will, a lavage, and then they will vomit up the plastic. And this gave us an opportunity to count how much plastic they've been exposed to, and then look at the healthier birds, not the birds that have died from plastic, the birds that have a couple of teaspoons or tablespoons of plastic and are still healthy weight and are in healthy condition. So we sampled the blood from healthy birds. They had to be above 100, had to be above 600 grams. Have I got time? Okay, I'm going to rip through it. So we did proteomics on it. We looked at the blood, and we can look for signatures in the blood. This is my second to last slide. Hang on, here we go. And we basically found all the organs were failing, even though it was a small amount of plastic. And really concerningly, we found signs that we have tested in Tasmania in humans. We found the same signatures that you find in the blood for dementia. So James Vickers over at the Wicking Institute found this signature for cognitive decline in elderly patients, a decline in your BDNF factor. We see a much greater decline in BDNF factor in the birds that are exposed to plastic. That's all. Sorry for going over time.